I think what we're going to do, I think I'm going to give you a quick few minutes on ligenesis, another quick few minutes on morphoceuticals, another juvenescence portfolio company. Faizan's going to give you an overview of Curate Bio, and then we'll chat a little bit like the Politburo up here. So apologies for the continuing American accents this morning. So I'm not going to belabor my background other than say you can read more about it there. I will just say, fundamentally, I am a drug developer, and in addition to my corporate role, I also tell folks that I speak regulatory. So I've had innumerable meetings with the FDA over the course of my 20-plus year career, and I look at that largely as currency of the realm, right? Because whether you're a 6-employee company or a 6,000-employee company, when you leave the FDA, at least in the US, with meeting minutes and a clear outline, or you get your IND, or I've taken all products all the way through to FDA approval, that is the same irrespective of company size, right? And so a lot of my work is focusing on advancing technologies through the drug development pipeline and ultimately to commercial success. So with that, very briefly, I'm going to spend just a few minutes telling you the most exciting story in biotech, at least as you might imagine that I believe it. Ligenesis came out of the University of Pittsburgh. University of Pittsburgh has a long history of innovation in liver transplantation, in particular because of Tom Starzl's pioneering work there to get the first liver transplant to be successful. My colleagues include Paulo Fontes, a transplant surgeon, as well as Eric Lagasse, my chief scientific officer, who discovered that the lymph node, which naturally acts, right? You have 400 to 500 lymph nodes distributed throughout our bodies. Naturally, they act as a bioreactor for T cells and help us fight infection. Without our lymph nodes, we would be unable to do so and would die and succumb to those infections. And just the way that cancer, when it's spreading, metastasizes and probes its environment for a hospitable environment, often finding the lymph node, right, and uses that as a bioreactor to form a tumor, we use that same fundamental biology, but instead of growing something metastatic and malignant, we grow a functional therapeutic tissue. And in fact, can engraft our cell therapy into a lymph node that will overtake that lymph node. The lymph node disappears, and what you're left with is a fully functioning ectopic organ that can exert life-saving effects. And we've shown that now over a decade's worth of academic work in my chief science officer's lab, rescuing animals from otherwise fatal diseases. So again, these tissues within the lymph node come to resemble their native counterparts. So in the case of the liver, which is our most advanced program, these are things with portal triads, central veins, right, bile ducts. This is tissue that histologically, if I take it to an expert, is indistinguishable from the native liver. And so based on that fundamental discovery that the lymph node can act as an in vivo bioreactor, and I'll say as a drug developer, by the way, in my past life working at like Amylin Pharmaceuticals, where we developed the first GLP-1 agonist for diabetes, there it was synthesized from the venom of a Gila monster, a lizard. And nature had acted really as our drug discoverer and had made that compound so potent that we could not artificially make it any better than it naturally was. And one of the things I've learned in my own drug development career is when you work with nature, you tend to be far more successful and much more effective than when you're trying to trick it or work against it. And here we're using the natural biology of the lymph node, combining it with the natural biology of the hepatocyte, which I mentioned earlier is nature's natural stem cell. And in particular on the business side of things, right, we have a number of things going for us. In the US, we discard almost 40%, actually more than 40% now, of the livers that are donated for transplantation are discarded. You cannot find a suitable donor. So what we do is we take that discarded organ, process it, engraft it through the mouth of the patient using an endoscopic ultrasound procedure. This is a low cost, the CMS billing code's of just about $2,000. Outpatient endoscopic procedure takes about 10 minutes. We engraft that into mesenteric lymph nodes adjacent to the liver where they grow and form these ectopic organs. Now, it turns out the liver is a hospitable environment for many different types of tissues, not just the liver, but also the thymus. And in fact, we have a very exciting paper coming out showing that we can take the thymus from a young animal, engraft it into an old animal, and both from a transcriptomic perspective, are you shifting that older animal to a more youthful phenotype? And right now we're dosing animals in a viral challenge, challenging them with an otherwise lethal dose of influenza A to demonstrate whether or not that ectopic thymus helps them survive that challenge. So again, these are functional tissues. We can grow an ectopic kidney for end-stage renal disease. Again, I'm not going to belabor any of this just because of time, as well as pancreatic islets also thrive within the lymph node and help to regulate otherwise diabetic animal. High level on ligenesis then, we're at clinical stage, our IND is open, patients are enrolling right now. We are in phase 2A in patients with end-stage liver disease. These are patients who are otherwise ineligible for transplant. They are relegated to palliative care and will die from their liver disease if nothing else is done. So we have enrolled the first few patients. We're waiting on livers to become available for them to process. Lead site right now is at Harvard's Mass General Hospital. Commercially scalable is something I just want to take a moment to talk about. So we're using discarded organs. This is a reservoir of cells that is easily available today that we can use. It only takes us 6 hours. It takes 3 people 6 hours to go from a donated organ to having the cells isolated, suspended, ready to engraft. Costs about $15,000 in reagents, right? So relative to Carte, to CRISPR, this is an incredibly efficient, very cost-effective procedure for the cell isolation. You combine that with the fact that you're using endoscopic ultrasound itself, a low-cost, minimally invasive procedure to engraft the cells, and you have commercially, right, the fundamental building blocks of a cell source that's readily available. You can isolate and suspend those cells at far less cost than you can other cell-based therapies, and then you can engraft the therapy in a way that's safe and also extraordinarily cost-effective. I will also say on commercially scalable, very briefly, that we've also demonstrated that a single site can serve an entire geographic region. So I can isolate cells at our facility that we've got up and running now at MGH. I can ship those cells anywhere in the Northeast, or I can ship the cell solution anywhere in the continental US for final processing. So this just goes to give you a sense, the commercial scale out of this therapy, a handful, 3 to 4 GMP centers across the US. I will not need a full sales force to detail this to every hepatologist in the US. A handful of geographic centers, you can service the entire continental United States. Low COGS, I will not belabor. That is just the fundamental building blocks of a good business. Relative to other cell-based therapies in particular, our margins on this therapy will be software-like. All right, and then lastly, if none of that appealed to you, I will just say on the fundamental supply-demand curve, right? The tragedy today is that dozens of people die every single day. Dozens of people will die today waiting for organs that will never become available to them. That is fundamentally because supply and demand works against the patient. You have one donated organ treats one patient. Using the ligenesis technology, because each single donated liver is composed of billions of hepatocytes, and we're only engrafting a few tens of millions or 100 million hepatocytes to form the basis, the seeds really, to grow these ectopic organs. Each single donated liver fundamentally disrupts the supply-demand calculus here, right? A single donated liver will treat 75 patients. Right, so I'm incredibly excited, both just on the basic commercial prospects, low COGS, incredibly scalable, and incredibly efficient from a supply-demand perspective, not to mention some incredibly exciting science. I didn't show you the pictures from our Nature Biotechnology paper, but when you see these ectopic organs, when you see a fully grown liver in an animal that's grown behind its knee, for example, just for the proof of principle, the lymph node, again, using that fundamental biology to really accomplish extraordinary medical therapies. And with that, I will mentally take just 3 seconds to now switch gears and tell you about another company in the Juvenescence portfolio, Morphoceuticals. I'm incredibly honored to be able to work with the scientific founders here, Mike Levin and David Kaplan out of Tufts University. I was just at Mike's lab last week. It is an impressive facility insofar as one of the main hallways, floor to ceiling, is journal covers. So if you're not aware of Mike Levin's work and David Kaplan's, I encourage you to check it out. And really, the best 15 minutes you will spend today, pull up Mike Levin's TED Talk. Do yourself a favor. It is one of the best TED Talks that you will see. 15 minutes. He literally leaves the main gentleman, whose name I now forget, interviewing him speechless in some of what Mike describes coming out of his lab. So this is highly regarded work, featured on the cover of journals like Cell and Nature and Science. Mike's thesis here is that if we think about the software of life and being able to reprogram the software of life, Mike's thesis is that that's not a metaphor. It's not a metaphor of reprogramming. Mike is actually proposing that we can literally reprogram the biology of life. He tells a story that is really quite remarkable, that if you look at the past few decades of remarkable innovation in medicine, he suggests that in many ways we're approaching this from a bottoms-up perspective. That is, when you try to manipulate the cell using gene therapies, genomics, metabolomics, CRISPR, it's the equivalent of computer programming in the 1940s, where literally, like you've seen the pictures, right? Like people unplugging cables and plugging them back in, because that's what it took back in the 40s to reprogram a computer. Mike's proposal is that that's really where we are at medicine today. We are reprogramming from the bottoms up. But if I ask you to take out your phone and to switch from email to Instagram, you don't break out the soldering iron, right? Because that's a highly inefficient way to change the hardware of your phone. You use software. And Mike's proposal is that, in fact, nature also uses software, and it is fundamentally a top-down control. It is a bioelectric control. That the software of life is fundamentally bioelectric. And so what he has shown is that if you understand the bioelectric signaling via ion channel and gap junction modulators of the cells, you can accomplish remarkable things that are simply impossible to engineer from the bottoms up. But if you take a tops-down bioelectric approach, you can. So what you're viewing here is a frog embryo through about 8 hours of development. This is not biochemical signaling. We're using voltage-dependent dyes here in Mike's lab to look at the electrical communication as the cells talk to one another to execute a program to build a frog. And it turns out in Mike's lab, you can manipulate and run subroutines within that program to make what's called a Picasso frog. If it looks like that frog has another arm growing out of its head, it does, because Mike told it to run a program to form another arm and grew one out of its head. Visiting Mike's lab is a fascinating experience. Mike has been able to program, for instance, gut cells to turn into eyes. He has actually grown animals with eyes in their tails, and they can actually quite clearly, through their behavior, see behind them. So you can induce bioelectrically cells to form an eye. It then recruits other supporting cells to form that organ. Again, from a bottoms-up perspective, what Mike is doing would be impossible. We are nowhere close today to being able to just understand the hundreds of genes involved that have to be turned on and off through precise timing to form a limb. But bioelectrically, everyone sitting in this room, I think, has two symmetrical limbs, because we do have an innate biological program to do remarkable things like grow organs and grow limbs. And if you understand that bioelectric program, you can tell the body to do just that again. And in fact, one of our lead indications is in limb regeneration. That's one of the low-hanging fruit from a regulatory perspective. I wonder if I can get that video to play. What do you think? Can you get that video to play? Okay. Oh, that's a bill. That's what it was. Yes. So this is by no means the best example, but these are animals that do not naturally regenerate limbs. You can cut off their legs. You could administer then in ion channel modulator 24 hours to the surface of the stump. 24 hours of exposure to that ion modulator to kick off the program. The program runs for 18 months and regrows a functioning leg that's innervated, that the frog can swim and stand on. And this is by no means the best example. This is just one example. We're now replicating and extending this work in mice, regrowing fingertips in mice with important potential implications for amputation stump health. That is just one application of this bioelectric technology. Lastly, I would just say, going way over, and Faizan's looking at the clock going, come on. Just very briefly, so in terms of the vision moving forward is something at Morphoceuticals we call physiomics, which is understanding this bioelectric program, understanding the voltage state of a diseased, of both the healthy organism and the diseased organism. And we use something called BETSI, the bioelectric tissue stimulation engine. It is an artificial intelligence program that looks at the healthy voltage state, looks at the diseased voltage state of an animal, and predicts which ion channels need to be opened or closed to switch the disease state back to the normal state. And using Betsy, we've been able to correct otherwise catastrophic notch mutations in brain development, so we can completely normalize what's otherwise a catastrophic birth defect caused by a notch mutation by opening a single ion channel, because Betsy understood in a way that a single human could not the transition of those voltage states between the diseased and healthy ones. So we're creating Betsy 2.0, which will be able to look at a variety of different organ types and use the software of life, really the bioelectric software, the programming, to change diseased organs back to their more healthy state. Obviously, potentially fascinating implications for aging here, which I will not belabor. So with that, and just lastly, so we are closing a seed round of financing. Again, tremendous work's been done. Kudos to Juvenescence for identifying this technology out of Tufts, by the way. My eternal respect for that. Mike and David are genuinely wonderful colleagues to work on. I've helped bring just some drug development focus to the work, trying to hit some clear drug development milestones to move it forward. With that, Faizan, a little bit on Curate Bio. Thanks.
We've got probably 5 minutes or so for my part, but what a fascinating, really fascinating story between Ligenesis and Morphoceuticals. So just to give you a brief background on myself, I've been the co-founder and CEO of Curate Bio, which we started a couple of months ago, really centered around the approach of consolidating a few different types of regenerative medicine assets. And partly, simplistically, when we think about it, there are probably two ways that you can think about regenerative medicine in, and one of which is the first being that you can take an in vivo approach to cell therapy, which is what Michael has described as, with the examples that you see with Morphoceuticals and Ligenesis. And then the other is an ex vivo cellular therapy approach, where we really try to interrogate how we can take what we call induced pluripotent stem cells or other types of technologies to create cellular medicines or live medicines that we can then transplant back into the body. And so it's really about the two coming together and really creating the sensational category of medicines or live medicines. Just a sort of a brief prelude to Curate Bio in terms of some of the assets that we have that we've been working with is we've got a company called Luxa Biotech, which is an RPE progenitor cell therapy, where we're in a phase 1/2A and we've enrolled about 5 patients so far. And we're showing where we can take, for a disease called age-related macular degeneration, we can transplant these cells, we can expand these cells we get them from cadaver eyes, expand the RPE progenitor cells, and then transplant them back into a patient where we're saying, like, for instance, what Michael's referring to, one of the pain points of regenerative medicine and realizing its full potential is really creating that sort of manufacturing paradigm to allow you to quite effectively and cheaply create live medicines. And so what this method sort of allows us to do is taking a sort of later approach for using cadaveric eyes, expanding out the progenitor cells, and then putting them back into a patient where we're seeing some dramatic improvements in terms of eyesight and sort of that whole tissue integrity being persistent. And then the two other programs that we've had so far are an iPSC-based Schwann cell therapy, where we can create functional myelin for, if you look at traumatic peripheral neuropathies or diabetic neuropathy and things like that. And there's another approach that we're working on that stems from using mRNA biology and having sort of a profound effect on reproductive medicine. And so those are sorts of technologies that we've curated within Curate. And the idea is to, again, create a sensational class of medicines where we can hopefully create cures, but as well as potentially be able to democratize sort of the manufacturing paradigm so that we can look into preventative medicines using live medicines as well. And that sort of might be, background. And so I wanted to sort of get us into a discussion between Mike and myself in terms of where the field is headed. How do we look at it from a regulatory perspective? How do we look at it from an investing perspective? And so maybe I'll start asking a few questions that I've jotted down that we can do with Mike. So as you know, Mike's had a tremendous career in terms of as a drug developer, and now he's sort of brought that expertise to what's very near and dear to me in terms of regenerative medicine. And so as a drug developer, how do you sort of evaluate early stage technologies?
Yeah, and can folks hear me okay? Do I need to? All right. One thing I wanted to follow up on one thing you just said, which is on the iPS cells. I think it's a great example of sort of, if you think about why now from an investment perspective in regenerative medicine. Like Genesis, we've taken a bird in hand approach we were talking about, that we're using donated organs today as a robust cell source. You have a lot of the natural biology of the hepatocyte as a stem cell. But fundamentally, right, that approach means it's allogeneic. It means you're going to have immune suppression. That's not trivial. It's trivial neither in terms of the cost of immune suppression nor in terms of the medical burden of patients who are quite ill, putting them on immune suppression. And so if you think about the next generation of therapies, right, using iPSCs, using those sorts of stem cells where you have off-the-shelf therapies that are not either immunogenic at all or as immunogenic, the ability to do what we're talking about doing at Genesis and at other regenerative medicine companies without the need for immune suppression, right? That is going to be a profound implication, not just clinically and on the medical side, but also just on the business side, right? Because it is still the case that you're fundamentally chained at Genesis to the donated organ. You have to wait for a blood type match, et cetera. To enable a truly off-the-shelf technology that does not require immune suppression, that's really something that only an iPS approach is going to enable. So we've taken that bird in hand, right? Engage the FDA from a CMC perspective, right? Genesis is the easiest CMC package I've ever put in front of the FDA. When you put a synthesized a xenobiologic like the venom of a Gila monster in front of the FDA, it's really complicated. When you're using human organs that we donate whole cloth, quite literally today, it's much easier. The iPS cells, right, are going to have a greater regulatory burden, right, from a CMC perspective, but that burden is going to be offset because of the clinical, I think, and commercial benefits as well.
Definitely. And I think we'll get closer to sort of, as we better understand developed therapies like Genesis and Luxa's, we'll get closer to sort of bringing down that cost for both autologous iPSC-based therapies as well as allogeneic iPSC-based therapies. There's a little bit of innovation that probably needs to occur to sort of to bring that type of therapy to the masses. But it's one that I think we'll have an interesting story to tell, 3 to 5 years from now, right? And then my next question, actually, and I guess we'll open up after this. We'll open up to the rest of the group. From a regulatory perspective, what can you do to sort of de-risk early stage assets? Yeah. And added on to that, is the bar higher for, say, a best-in-class therapeutic versus a first-in-class therapeutic?
Yeah. No, those are great questions, and having met with the FDA on a lot of different occasions, I'm so struck that working with a number of brilliant academics spinning just incredibly innovative things out of the lab, right? There's also a large disconnect between understanding that fundamentally it's a little bit of a stretch, but not much, to say the regulators, as long as you can tell a story about why you think it might work, that's enough. Right? You don't need to understand the way that the academic desperately has this thirst to understand the exact biochemical signaling of each part of that pathway, and that's how you build an academic career, and that's great. As long as you can tell a story to the regulators that's plausible on the efficacy side, you've sort of checked that box. Conversely, on the safety side for regulators, right? If you think of like a teeter-totter, right, of like safety and efficacy, The fulcrum of the teeter-totter from the FDA's perspective is way skewed to the side. Any whiff of a safety signal, and you just have a world of concern, right? Where for many folks in academia, that maybe not, they don't focus a lot on dose ranging. They don't look at off-target toxicities because they're trying to get the scientific publication. Those things become absolutely central. And so being able to demonstrate to regulators early on, not so much whether it's best in class or first in class, you can pretty easily check the box with the regulators. Here's why we think it works. The key is being able to demonstrate to them, here's why we think it's safe. And if they believe that you're putting patient safety first and you have a robust safety story, then you can have a very productive dialogue with the FDA. If you go in thinking they're going to be enamored by your science paper that you just published, yeah, they really could care less, right?